Hi everyone, welcome to my channel Pure Biology. Today's topic is the Lac Operon. Okay, so before going to this topic, I just want to say that I have explained different topics related to the biological science. Okay, so you please check out that videos and. Obviously this is a new chapter or new topic, that is from genetics, and before this topic I also explained different topics related to genetics obviously in my previous videos, and you obviously check out these videos. And also I request you, if this channel is helpful to all of you, please share with your friends and obviously subscribe my channel to grow our channel, because it is very new channel, and I will be very grateful if you subscribe my channel. Okay, so let's start. This is our topic, today's topic, that is the Lac Operon. What is the Lac Operon? Lac Operon actually is an operon. It is an operon unit of bacterial gene expression and regulation, which includes the structural genes and the regulatory sequences recognized by the regulatory gene product. So Lac Operon actually is a unit of the bacterial gene expression and the regulation that includes two kinds of genes. One is the structural genes and another is the regulatory genes. Okay, the structural genes, within the structure, these are the three structural genes, Z, Y, A. And in case of Z, this actually binds one enzyme. That enzyme is the beta-galactosidase. Y is the another structural gene that binds one enzyme, that is the permease. Okay, and A is another structural gene that also binds to the trans-acetylase. So these are the enzymes that bind to these three structural genes. Okay, so another also important factor, obviously, the function of the Lac Z, Lac Y and the Lac A. Lac Y and the Lac A. So what is the function of the Lac Z? Lac Z actually, it helps to dissociate the lactose into glucose and galactose. Okay, glucose and galactose. And lactose has another function. It also Lac Z has another function. It also transform or convert the lactose into the allolactose. This is the function of the Lac Z gene. Another function of the Lac Y, that is actually it binds the enzyme permease, and its function is it actually helps to transport the lactose into the cell. Okay, it helps to transport the lactose into the cell. And the third gene, that is the Lac A, it helps in detoxification of the compounds by transferring an acetyl group from the acetyl CoA to the beta-galactosidase. Okay, what is the function of the Lac A? Lac A is the gene. Its function is it actually transport the acetyl group. Okay, it transport the acetyl group from the acetyl CoA to the beta-galactosidase. So these are the functions of the structural genes. Now we will learn what is the function of the regulator gene. The regulator gene actually here is the I. Okay, this is the regulator gene, and its product is the regulatory protein. This product is the regulatory protein here, regulatory protein. And what is the function of this regulatory protein? Actually, this regulatory protein, this protein binds to the O. O means the operator. Okay, this is the operator, and regulator protein, this binds to the operator. When it binds to the operator, actually it's block the promoter. Promoter is the actual binding site of RNA polymerase. Here, this is the binding site of RNA polymerase. If the regulator protein or the repressor protein, that is called the repressor protein, this repressor protein binds to the O operator, then the RNA polymerase, which generally binds to the promoter and helps to transcription and translation, continue process, this totally prevented. Okay, the binding of RNA polymerase to the promoter is totally prevented. So here, what is the Lac operator? We learned that what is the Lac operator. This is actual region. Region is lie between the promoter and this structural gene Z. Okay, promoter and the structural gene. and interacts with regulatory protein which controls the transcription of operon. Clear? It controls. It is the one binding site where only regulator protein or the repressor protein can bind this site. Clear? And this is the very short region and only the CIS, okay, this is the cis site of the DNA, okay, this is called, cis site, cis acting site of the DNA, and regulator protein or the repressor protein that is produced by the regulator gene or the I gene, this bind only the cis site of the operator, okay, it binds only the cis site of the operator. So, Lac repressor bind to the, and what happened after that? When the Lac repressor bind to the cis site of the operator, then what happened? It prevents the RNA polymerase. Okay, the activity of the RNA polymerase, and RNA polymerase cannot bind to the promoter region. Clear? So this is the operator. Next one is the Lac repressor. What is this Lac repressor? Lac repressor actually, this is the trans-acting regulatory gene. Okay, the product of its product actually the trans-acting regulatory gene. Okay, and that regulates the transcription of the structural genes. So what is the function of the regulator gene? Regulator gene's function actually it controls the transcription process of the structural genes, and the regulator gene is a gene that codes for the protein involved in regulating the expression of the other gene. Why it is called the regulator gene? Because it controls the expression, or it helps to regulate the expression of the other gene, and that's why it is called the regulator gene, and its product is called the regulator protein. So this is actually here is the I. I is the regulator gene and its product is the repressor. Next one is the promoter. What is the role of the promoter here? It is the recognition sequence for RNA polymerase. This site is very important because RNA polymerase bind here and code for any product. It is not code for, it does not code for any product. That means without RNA polymerase, any product that is formed cannot bind here. Okay, cannot bind here. So the effect of P mutation confined to the genes that it controls on the same DNA strand. If the mutation is possible or mutation is occurring in the promoter gene, that remain within that DNA. Okay, its control or its activity that remain within that DNA. So this is the promoter. This is Lac repressor, this is promoter, and this is operator. And these are all the structural genes here. So you can understand obviously what is the structural genes: Lac Z, Lac Y, and the Lac A. Lac Z, its function that it converts the lactose to allolactose or glucose and galactose. And Lac Y, it transports lactose to the cell, and Lac A actually it helps to transformation of the acetyl group from the acetyl CoA to the beta-galactosidase. Clear? And its enzymes are that. First one's enzyme is galactosidase, beta-galactosidase. Second one's enzyme is the parmease, and the third one's enzyme is the transacetylase. Clear? So these are the enzymes that bind with these structural genes, and these are the other regions that are very important for the Lac Operon. So this is actually the topic I very briefly explained about the Lac Operon. Its next part I will discuss you in my next video. Okay. So please you again I say that please you subscribe and obviously share my channel. Thank you.